for Brazil. That's Nova Analytica. They're located in Sao Paulo. But that's regarding the sales part. But we also have a really strong cooperation network with a lot of academic institutions. Here's just a few of them. So we try to be really a partner also for science and for a lot of academic institutions. And together with this, I'll do the laser pointer here. You can see on the right-hand side something very special that we launched this year. It's called the Kuhner Science Fund, and that's an initiative that should make it easier also for academic institutions or startups, for instance, to purchase really the high quality shaking equipment. And this was an initiative actually by our former CEO. He's on this picture down here. As I said, we are a small family-owned company, and this is our former CEO, Markus Kuhner, who guided the company for about 45 years. And this year, his daughter, Laura Kuhner, she took over as CEO. So, with this transition, we launched this science fund. As said before, our main business is actually incubator shakers, and here's just a few of them. So as you can see, for instance, there's incubator shakers with a hood around. So these ones can be, for instance, temperature controlled, CO2, oxygen, humidity controlled. But in the portfolios, there's also benchtop shakers, and also really huge shakers with a lot of different trays. So we can shake a great variety of shake flasks and microtiter plates. And down here in this corner, you see a shaker which is called MPSC Automated, and this is our latest launched product, which is a fully automated microtiter plate shaker. So now it's moving. Just a few remarks regarding the shakers, what might be interesting for mammalian cell culture. So the interior is fully made of stainless steel and very hygienic design. And what is kind of unique actually is that all these shaker equipment, it's made with an encapsulation of foam. So they are really well insulated, which leads to extremely low condensation problems and a really nice temperature distribution. So that's kind of a unique point regarding these shakers. And what is also very special is the drive we are using. It's a direct magnetic drive. So as you can see here, there's no gearbox, no belts, nothing that can break. So it's a really robust technology that we are using in these shakers. And down here, as said before, these days the shakers are also ready for automation. On this picture here, you can see this microtiter plate shaker, which can automatically present microtiter plates for further processing. But this is just to show you what we are mainly doing. Another interesting topic, and that's what you've seen on the first picture when you saw the big group of people. That is actually the orbital shaken bioreactors, which is quite an interesting type of bioreactor, quite unusual actually. And as you can see here on these pictures, these orbital shaken bioreactor, they are basically we could say big, huge shake flasks. Yeah, it's just a big vessel, no stirrers in there. So they have the same hydrodynamics, for instance, as a shake flask or microchiter plate. So it's very, very easy to scale up. And as a contrast to stirred tank bioreactors, they have really, really low hydromechanical stress because there is no stirrer in there with the tip. So a bit of a niche product actually, but very, very nice tool for problematic scale up or for cells or processes where hydromechanical stress is a big problem. Just to show you one example here, which was done with Zurich University of Applied Science. They, for instance, did chocolate. So the cultivation of cacao in a 50-liter single-use orbital shaken bioreactor. And on the right-hand side, I just wanted to show this this video. This is actually the largest orbital shaken bioreactor in the world, and that's a 2,000 liters or 2 cubic meters of orbital shaking. And just to give you an idea how huge shaken processes can be these days. Yeah, so this is our CEO down here, and that's a vessel of about twice the size of a human. All right, so with this, that's enough regarding products. Let's start with the actual scientific part of this talk. Coming to my field here, which is the different online measurement tools for off-gas analysis. I think off-gas analysis is quite well known when it comes to larger bioreactors, but not so much for shake flasks and microtiter plate experiments. And why is it actually so important? So just looking at this overview here, we all know that our cells, they need a well-balanced medium. Yeah, they need carbon sources, nitrogen sources, and so on. But in any process, we also need oxygen. So oxygen is sort of our universal substrate, which is not that abundant in the liquid broth. Yeah, but it's very essential to any shaken process or also any stirred process. And then on the product side, of course, we have cells that are produced, or we have our main products like proteins or microbial fields. There's also a lot of other products, but there's also CO2, yeah, which is coming from any process. So I think we can say that the oxygen is sort of the universal substrate, and CO2 is sort of the universal product in our processes. And now, what could now be interesting? Actually, that's the what we want to know about our cells. It's the oxygen, what we call the oxygen uptake rate, the OUR. That's the amount of oxygen that the cell is taking up, and the carbon dioxide evolution rate. That's the carbon dioxide that the cell is producing. But we are not that lucky. So in a shake flask or in other processes, what we can measure is the oxygen transfer rate and the carbon dioxide transfer rate. So not exactly on the cell level, but what is transferred between the liquid and the gas phase. But we are very lucky, and because of the solubility of oxygen in the medium is really low, the transfer rate and the uptake rate of the cells, they are very much alike. And it's slightly different for the carbon dioxide, because carbon dioxide can also be stored in the medium. And it's heavily influenced, for instance, by pH changes in bicarbonate buffered systems. And then there is also the quotient of these two, which is called the transfer quotient, and that's the quotient of carbon dioxide transfer divided by oxygen transfer. And that's a really nice fingerprint of the cell's metabolism, actually. So why actually oxygen transfer rate? Why OTR and not just dissolved oxygen? Let's try to put down here what are actually the main differences. So the OTR, it's already in the name, it's a transfer rate, and the unit is millimole oxygen per liter of liquid per time. And it's stating how much oxygen is taken up by the culture medium. And this means that it's also stating how much oxygen is actually taken up by the cells. And if we look at this, quite easy equation down here, we can see that it's very well connected actually to the amount of viable cells. It's just connected via the cell-specific oxygen uptake rate. And on the other end, for instance, thinking about reaction stoichiometry, so when glucose is consumed, this oxygen consumption of the cells is also very well related to how much glucose is taken up. And on the other hand, when we think about dissolved oxygen, this is a concentration reading. It is also a result of the respiration of the cells, but of course also of the shaking conditions, of the solubility in the medium, also of the pressure, of the headspace, gas concentration. So it's influenced by a lot of different parameters. And I think one can summarize a bit like this. So when thinking about these transfer rates, oxygen transfer rate, carbon dioxide transfer rate, we get a feeling about what the cells are actually doing. Yeah, it's a consumption or a production rate. And when thinking about dissolved oxygen as a concentration, it is more telling what the cells are actually experiencing in the culture growth. So I think we can say that, or it seems to be that OTR and CTR are quite a valuable measurement, but how actually to determine in a shake flask or even in a microtighter plate. And for this, we made a little trick, let's say. So we're not measuring directly actually what's going on in the shake flask, but we introduced a little off-gas recycle by introducing a little microfluidic pump and oxygen and CO2 sensors. In this way, we are able to measure what's going on in the shake flask. And this is all overlaid by another flow of gas, because you can imagine if we would just have it like this, then the cells at some point would be starving from oxygen because no fresh air can come in. So this is overlaid by a fresh air line. And now the measurement principle, how does it actually work? So in this graph, it's just schematic. We can see the oxygen concentration and the CO2 concentration. And with such a device, we can now stop the inflow of fresh air. And then we see the oxygen decreasing in the shake flask and the CO2 increasing in the shake flask. And from that slope, we can directly measure the oxygen consumption of the cells and the carbon dioxide production. And after this measurement, the airflow of fresh air is opened up again, and the system is going back to normal. So we're introducing these direct measurements. And now the tricky thing here is this was first invented actually for microbial cells, but by having this measurement principle, it's quite easy to make this measurement longer if you have cells with really low respiration. Yeah, and this is what we also published this year. So with this system, we can really adapt also to to mammalian cells and have a really high measurement accuracy, even on the shake flask level. And now in the following slides, I would like to introduce you to some application examples that have been reported in the last years. So the technical realization of such a system then looks like this. So all the sensors and the pumps, they are included in this box here. And as you can see from this picture, now basically any shake flask can be connected to the system. On the left-hand side, it's more for the microbial applications. It's glass flasks, different sizes. Also different shapes can be connected. So and on the right-hand side, you see single-use flasks, as they are quite usual for mammalian cells. And now the idea is that you don't need to have a special flask or a very special connector to monitor these flasks, but there is an adapter, which you can see here on the right-hand side, that is, can be screwed just on top of a standard single-use flask, and the sterile barrier of this flask is not touched. So we're just introducing this chamber above the shake flask, and then we are measuring the concentration and the concentration gradients in this chamber above. So it's a standard single-use flask, and the sterile barrier can also stay attached. So that's the technical realization, and now let's have a look how it looks in actual in an actual run. So just put this here to have an introduction to how the data looks like, and we will then go over to published data. So what we see here is the oxygen transfer rate on the y-axis over time on the x-axis, and this is just the standard CHO cultivation in a small Corning flask. And what we see here in the beginning, so first 3 to 4 days, we see some exponential increase in the oxygen uptake of the cells. And then in the later stages, you see here there's a first little bump, and there are more later on. And then at the end of the cultivation, you see a really steep decrease in the oxygen uptake. Yeah, so this is kind of a typical behavior that we see here. And now going over to this publication, well, some data that was published last year, actually with the antibody-producing cell line. And here on this first graph, you see the red line. That's again the oxygen transfer. So pretty much the same shape as we saw on the slide before. Also some exponential growth in the beginning, and then they just also describe this as really distinct phases that we can see here. So again, this phase where the oxygen uptake still goes up, and the last phase where the oxygen uptake is really small after the steep decrease. And one thing we can state here is actually that we already see from the online signal when the antibody production stops. So in the moment where the oxygen uptake really crashes, and also the antibody production stops. And of course, also viable cell count was measured here, and we can see that there is seems to be a really strong correlation between the viable cell count and the oxygen transfer, which also makes sense that let's say, as a first assumption, twice the amount of cells would also roughly take up twice the amount of oxygen. But what you can also see here is that the oxygen transfer that we are measuring, it's always a few hours ahead, actually. So when you look up here at the green graph, you see that there are still viable cells available here, but the oxygen transfer has already crashed. And when they measured metabolites or carbon sources, we can also find some really nice correlation here between the main carbon sources. So the first bump here in the oxygen transfer is very well correlated to the depletion of glutamine. Yeah, that's the first dashed line. The highest peak here was always when the glucose was depleted, and then the sharp decrease here was at the point where the last carbon source was depleted, and that was when actually lactate was consumed. So just by looking at the OTR in a shake flask, you can actually read quite a lot. So what carbon sources or nitrogen sources are depleted at what point, a rough estimation of the viable cell count, and also when the antibody production stops. So quite a few information just from a single run here. And later in that publication, they used these correlations that they could draw from these first results to check how actually the cells are influenced by different shaking conditions. And different shaking conditions meaning different energy dissipation rates in terms of or being representative for the shear stress or the hydromechanical stress that the cells see. So they took different shake flask sizes, different filling volumes, different shaking speeds, and they could see just from looking at the oxygen transfer, which was well related to the viable cell count, what are actually the conditions that are stressing the cells and leading to slower growth and also to a much longer cultivation time. So in this case, they had sort of let's say a soft sensor of cell viability just via oxygen transfer rate measurements. And the last step, they also correlated then the antibody production, which was actually quite unaffected by the shaking conditions, only for the very high shear stress and the very, very long cultivation time, decrease in antibody production was seen. So that was the first application example, and was mainly focusing on oxygen transfer. And we haven't heard anything about the carbon dioxide transfer so far. So that's my second example here. In this graph, we see in orange the oxygen transfer rate, which looks similar but not totally the same as in the example before. So also some exponential growth in the first phase, and then some slow decrease in the later stages. But what is really impressive here, actually, when I saw this the first time, is the carbon dioxide transfer, which is much, much higher than the oxygen transfer in the first phase of the run. And in the later stage, it is lower than the oxygen uptake. And why is this? So when just looking at the simple, very, very simple stoichiometry, when a cell is using glucose, just using for energy production, so just burning glucose, we would expect this reaction stoichiometry, So we would assume that the same amount of oxygen is needed or is taken up as carbon dioxide is then also produced. So we would, that would be a good first assumption. And also when thinking about the conversion of glucose to lactate, that's where no oxygen or CO2 is actually needed or produced. So just from looking at this, we would assume actually that the OTR should be pretty much like the CTR. But when lactate is produced, we get protons and the pH is going down. And now being in a bicarbonate buffered system, this will make the buffer react to the CO2 side. Yeah, so every time the pH is slowly dropping in our cultivation, actually CO2 is released from the buffer. And that is what we see here in the first phase, when a lot of glucose is present, the cells are producing lactate, pH is slightly going down. Then we see this CO2 overproduction. And in the later stage, when the lactate is consumed, it's vice versa. So we can see from this data that the carbon dioxide, because of the strong or the high solubility in the medium and the bicarbonate buffer system, it is not just the cell respiration that we see, but it's also the reaction of the CO2, which is stored in the medium. So it's always a mixed signal, we can say. But by knowing this, in this publication from 21, this was used to actually predict the lactate formation just from the difference between carbon dioxide production and oxygen consumption. And this was really a perfect match in this case, really nice data. So just from an online measurement in a shake flask. So I hope that with the last two application example, I could show that there is quite a strong correlation actually between OTR and CTR and viable cell density, also glucose consumption, and the formation and consumption of lactate. And that made us think about a soft sensing approach. That's the project we are currently working on, so that's more of an outlook actually, where we are trying to build a hybrid model, which is half mechanistic and half machine learning algorithm, and that is using the OTR and CTR data to actually predict what's the current viable cell density and how much glucose is still left in the medium. So that's some ongoing work here. So what we've seen so far, that's actually on the shake flask scale, where you can imagine that there's still enough space for oxygen and carbon dioxide sensors. But just recently, we launched a product that can measure oxygen transfer on the microtiter plate scale. It's not able to measure carbon dioxide, but oxygen transfer. And the working principle here is actually it's pretty much the same as we've seen before for the shake flask. So what you can see here on this lower picture, it's like a little box where a microplate can be put in. And above the microplate, that's what you see on the little schematic here, there are 96 tiny little fluorescent optical oxygen sensors. So it's measuring the oxygen concentration in each individual well of the microtiter plate. It's not just the sum of all wells, but it's the really individual in each well. And the measurement principle is similar to the shake flask, because the microtiter plate, it can be lifted with a pneumatic mechanism. Just, I hope you can see on this very small video, it's going up and down. It takes longer in real life, so this is just for show here. And when it's pushed up, then we have the same as before for the shake flask. Then the oxygen partial pressure in the well of the microtiter plate goes down. And this is then again converted to a oxygen transfer rate reading. So same principle. And one last application example here. So it's also done with a 96 well microtiter plate, also done with 2 different CHO cell lines. And in this application, people did kill curve experiments. So just checking about the effect of different pyromycin concentrations on the growth of the CHO cells. And they were able to put 11 different antibiotic concentrations here. And on the right-hand side, you can see the oxygen transfer rate readings for those different pyromycin concentrations and the effects on growth. And again here, in the upper graph, you see the oxygen transfer rate. In the lower graph, you see the viable cell densities. And again, they correlate very well, and they could really find the threshold when the pyromycin is most effective here. So with this, I would like to come to the end and just sum up a little bit here. So I hope I could show the value of OTR and CTR readings in shake flasks and also in microtiter plates, and also a bit the difference to dissolved oxygen. We've seen that those measurements are able, or we are able to do those in shake flasks and microtiter plates. And we've seen also a very strong correlation to the viable cell count. Oh, that's wrong here, actually. Of course, to the oxygen consumption. No, it's for the glucose consumption and also the lactate metabolism. And with this, I would like to thank you for your kind attention. And yeah, if you're interested in those slides, I hope this works. So this is a QR code, and we're happy to take a photograph, and that's how we can get in contact, and you can request the slides from me. And now I think there is still time, and I'm happy to answer your questions.
I hope you can see us.
Yes.
Yeah. If you
I see the audience. Yeah.
Everyone has taken the picture. Otherwise, I would suggest that you stop sharing the presentation so you can see us. Yeah, big.
Yeah. All right then.
Yeah, we do have about 5 minutes for questions. Who wants to go first? Okay.
Wait. It's the expert in oxygen. Me first.
Okay. So we are debating who.
Okay, thank you.
Thank you very much, Andreas, for the nice presentation. I enjoy a lot this field. You show some measurements of O2 and CO2, and CO2 is normally a very consistent and reliable measurement, but oxygen changes and drifts. And in your system, if it loses calibration, I have to send to Switzerland, or can I have? This is a problem we have here in Brazil. We have the blue sands, and we got chocolate instead. So we have problem with the maintenance of the O2 measurement. How can we do this with Switzerland?
Yeah. So The oxygen sensor and the CO2 sensors, I would say they have a, let's say a medium lifetime of roughly 3 years. Yeah? And within this time, the oxygen sensor has a self-calibrating mechanism. So I can't now explain in detail how it exactly works, but the oxygen sensors does not have to be maintained until it comes to the end of its lifetime. And the CO2 sensor, there is a calibration procedure that you could do in your lab. For instance, if you have a incubator shaker running at 5% CO2, once in a while you can adjust the sensors that they go back to this value, so it doesn't have to be sent somewhere. And so the maintenance until it comes to the end of the lifetime can be done in the lab and doesn't need a technician.
Well, thank you very much for your very nice talk. I have 2 quick questions. One is related to the effect of shear protecting agents, for instance, for cells that are in suspension, like Poloxamer 188. Do you have an effect of these types of agents in your measurements?
I actually have not seen, I have to admit, I have not seen data for mammalian cells yet. We're working on a different project at the moment where we are checking baffled flasks, and that's where we are working with this antifoam agents, for instance. And there we don't see an effect on the measurement. So the measurement is not influenced. Of course, we see an effect on let's say on the cells or on the foam formation. That's why we are measuring different values, but it's not influencing the measurement itself.
Oh, okay. No, that's
good.
good.
Answering the question?
Yeah, no, that's excellent. And another question is regarding cells that need to be adhered, for instance, to microcarriers. Would this system also work with microcarriers?
Actually, nice question. It's also a project we're working on at the moment to show this the first time. But that's actually a nice thing about off-gas analysis. Yeah. So as long as you're measuring in the liquid, or you need to detach cells from the microcarriers to measure something. Yeah, but the off-gas, it doesn't really care if the cells are in suspension or if they are attached to microcarriers. So it doesn't really matter, yeah. So because also the cells on the microcarriers would need the oxygen transfer to go from the gas phase to the liquid, and that's why. What is the difficulty, of course, is that even on microcarriers, you would probably have a lower cell density per volume, let's say, than compared to a suspension cell culture. And that would give lower, let's say lower respiration activity, and that's why it would give a lower reading, yeah, which makes it more difficult at some point. And that would be when you need to make this measurement longer again to adapt for this.
Okay, yeah.
So at the moment we're trying this for stem cells, adherent growing stem cells on microcarriers, for instance, and that's also working.
Yeah, that was exactly my question. If the method was sensitive enough to get these smaller variations. Thank you very much. Yeah.
Welcome.
Thank you, Andreas. I have one last question. I was really impressed with the 2,000 liters bioreactor shaking. Never saw something like that. So it doesn't have an impeller. It shakes to provide mixing. Does it have an asperger to provide oxygen, or is it just surface aeration?
It's just surface aeration. So same principle as in the microtiter plate, just huge. Yeah. Of course, like KLA values are going down the higher you go, yeah, the bigger you go. But it's still very sufficient for mammalian cells, and there are a few productions on industrial scale going on like this. Yeah, so it's still high enough oxygen intake just by the surface aeration in the big 2,000 liter.
Okay,
Yeah.
awesome. So thanks a lot. Oh, we have one last question. Sorry, one last question.
Hi. Thank you very much for the presentation. I have a briefly question. How do you measure the energy dissipation rate to evaluate the influence of the growth cells on the growth cells?
So I have not measured it, so it was in this publication. But I know from that institute they have special equipment to determine the energy dissipation rate for a single shake flask. So by measuring the power input, and there are different correlations for the maximum energy dissipation rate depending on the power input. So that's I can't exactly. It's like a torque measurement, measuring what's the power input to the single shake flask. That's the basic principle, let's say.
Okay, and did you do this in shaking bioreactors too, or just in flask?
We do have some data for the orbital shaken bioreactors. I'm not fully sure. Yeah, but there's also some publications about this where these correlations are found. So how does it scale to bigger flasks? It's something. Yeah, I don't have exactly in mind, but there is a publication dealing with the power input on large orbital shaken vessels. Yeah.
Okay, thank you.
So if that's something you're interested in, we should just get maybe in touch, and I can look this up for you.
Thank you.
Welcome.
All right. I guess we're done. Thank you very much, Andreas. And yeah, we will contact you for further questions. Have a nice afternoon.
Thank you very much.
Yeah.
Have a nice lunch. Bye.
Finally, it's time to eat. Yeah, we have food again like yesterday here and the tender.